Hello, and thank you for tuning in for Science in the News Spring Lecture Series through our online live stream. I'm Kelsey, co-director of Science in the News, and tonight you'll be hearing about In the Loop with Poop, Microbes in Health and Immunity with graduate student Chris Garris. We'll get started in just a couple of minutes, but please participate in the lecture by asking questions using the link above. At the end of the lecture, please give us some feedback using the form below. We will answer most questions during the lecture, but if we don't get a chance to answer your questions, we'll do our best to send you an email following the lecture. So please be sure to include an email in your response. Thanks for being with us, and we'll get started in just a couple of minutes. Hi everyone, we're going to get started. Welcome to the Science in the News Spring Lecture Series. I'm Kelsey, one of the co-directors of Science in the News, and this is Vinny, the other co-director. Science in the News is a graduate student group based out of Harvard University, and it encompasses all of the Graduate School of Arts and Sciences. And our main mission is to provide events and opportunities to bring scientists and non-scientists together. And the lecture series is just one of the ways in which we do this. Our lecture series, we have a fall and a spring version. Our fall one many of you have been to, and it occurs at the Longwood Medical Campus by Children's Hospital. And our spring series, of course, occurs here. So Vinny's going to be telling you more about the spring series and how tonight's going to work. But just to tell you about some of the other events that we run, we also have an event called Science by the Pint. This event is really fun, casual, and informal. It takes place at the Burren in Davis Square, which is a pub. We invite a faculty, scientist, and members of their lab to join us. And the faculty member gives a brief talk about what they do, and then they and members of the lab mingle table to table and answer your questions over good food and beer at the Burren. And so we have an event coming up on Monday. You might have seen the slide come across the PowerPoint. That's with Dr. Chris Garris, and the theme is astrophysics and computational mathematics. And I'm sure he'll make it sound less intimidating than that title. So check that event out if you can at 7 p.m. on Monday night, and those happen the second Monday of every month. We have a new schedule which will be up online tonight, and we also have new flyers that will be available next lecture. In addition to this, we also have an online newsletter which I encourage you to check out on our website. It's called Signal to Noise, and we run many other events, but I'll let Vinny take it away.
Awesome. So just to give you a sense of how tonight's lecture will work, if you've been to these events, you already know. So what will happen is Chris will be giving our lecture today, and Chris is actually a second-year student in the immunology program, and he studies the interaction between the immune system and cancer. And he also studies the role of the microbiota, which is what he'll be talking to you today about. So the format of the lecture actually works in that Chris will give a part of his lecture, and then he'll stop for questions periodically. So we ask that you please hold your questions until then, and we'll have a short intermission halfway through Chris's talk as well. And if you haven't already, you should grab one of these handouts, which has a nice handy term sheet, as well as a little abstract for tonight's talk, and a survey, because we would love to have your feedback as well. And I'd like to also take this opportunity to thank our sponsors. the Graduate School of Arts and Sciences, Harvard Medical School, the Harvard Coop, and the Biomedical Graduate Student Organization. And with that, I'll turn it over to Chris.
Two microphones. So, hi everyone. I'm your lecturer for tonight. My name's Chris, and I'm a, as Vinny mentioned, a graduate student in Harvard's immunology program. And my talk tonight is titled "In the Loop with Poop: Intestinal Microbes in Health and Immunity." So, first we want to clarify, what is the loop in this context? So, the loop in this case are the intestines, both small and large. And these are really known as absorptive organs for nutrients and water. The small intestine for absorbing nutrients, and water for reabsorption through the large intestine. And the curious thing about these structures is that they're home to a ton of microbes. So the second part of the talk title is poop. So why poop? Well, for one thing, as you can tell by the smell of said object, it's full of microbes. Actually, like 60% of the dry mass of poop is bacteria. And one thing that's nice about this, though, is that it transits through the colon and also through the small intestines. So basically, it goes through your intestines, and it can provide more or less a picture of the environment that exists within there. And also, it's very easy to collect. So I'll go through our plan for the evening with a few main points, basically questions at this point. So first, we'll start off by, what is a microbe? And then, where do these bugs live? I'll be using the term bugs for microbe interchangeably. And why are these bugs important to health? And then more about the research side, how we study these bugs, and what I specifically am researching. So from here, we'll take our magnifying glass into the gut and the bacteria that reside inside. So to start, about definitions of what is a microbe, it is, as it would imply, a small organism. So this can include a diverse range of life. Here is a tree of life, for example, with different types of bacteria, archaea, and eukaryotes, the three main kingdoms of life. And a microbe can exist within any one of these, actually. So principally, we know most about bacteria, but that doesn't exclude the influence of certain fungi that could exist within the gut, or even archaeobacteria as well. But for the most part, I'm going to focus on bacteria within our intestines. So we know that most bugs live in the gut. That doesn't mean that they only live in the gut. They live actually at any site that's exposed to the outside environment. So there's bugs and bacteria on your skin, in your nose, in your lungs as well. But it seems that the majority of the species that we know about reside within the gut. So here's another diagram of this. And they're present actually in both the small and large intestine in this case. Though there actually are more bacteria present within the colon, which would be the large intestine in this case. But as I mentioned before, any site that has exposure to the outside environment can be a reservoir for bacteria. So I'm not specifically excluding them in this talk, but I'm going to focus on what we know from the gut, because that's where most of the research lies at this point. So why would a bacteria want to live in the gut? Well, for one thing, there's access to nutrients, in this case, dietary fiber in the form of carrots and celery. And this dietary fiber, actually, with the chemical diagram shown here, is basically a long carbohydrate, can be fermented by bacteria into these little compounds called short-chain fatty acids. So basically, this is a byproduct or a metabolite generated by bacteria that can actually be used by the host for certain circumstances. And we get into that further. And also for energy as well. So these bacterial byproducts actually can promote a healthy gut. So how do we know that bacteria are even important for health? So there's these curious, you could call them almost freaks of nature that we have that are called germ-free mice. And they really make the point hit home that bacteria in the gut or anywhere on the body is actually rather important. So these mice are actually completely germ-free. And I'm going to, for the purposes of this talk, have the dark mouse as the germ-free, and a conventionally raised mouse, which is a mouse that lives a normal germ-existent lifestyle, as the white mouse this way. So the germ-free mice are raised in these things called sterile isolators from birth till death. They are essentially bubble boys in the sense that they're never exposed to any kinds of bacteria or fungi. So they're completely clean. So if you take a look at their intestines with a cross-section right here, you would see that there would be no bacteria within the gut lumen, which would be the side that would be where you'd have food and nutrients in. And this would be host tissues. Whereas the conventionally raised mouse that's not raised in a sterile isolator would actually have an abundance of bacteria present within its intestines. So using these models, you can infer that certain types of bacteria or the presence of bacteria can be important for host physiology. So there are some numerous problems that are wrong with germ-free mice. And I'll enumerate just 3 of them for now, but these are not the only problems with them. So first, they have some pretty serious defects in their immune system. And they are very susceptible to infections. And they have some curious metabolic changes that occur within them. And this is a sad germ-free mouse. So just for a brief overview, I'll just do a view from 10,000 feet of the immune system with basic principles. So the immune system, its more or less primary goal is to protect against disease. And it does this by having mechanisms that can distinguish self between non-self actually. But this isn't always the case because there are certain conditions called autoimmune diseases where your immune system can be directed against yourself. So, but for the most part, it usually holds up this model pretty well. And also, it's an interaction of many different cell types that happen. So these can include different cells like a macrophage right here, a T cell, this cell called a neutrophil, a B cell, and a dendritic cell. So I'm going to focus actually on the T cell in this context and how it's affected by a germ-free environment. So we can see that certain types of T cells are actually absent in the intestines of germ-free mice. So if we have this cell called a Th17 cell, which is important in, I guess, barrier functions and barrier immunity within the intestines and also the skin, in a germ-free mouse, you see that their numbers of Th17 cells are very, very low. But when you add back, specifically, you feed them this bacterium called segmented filamentous bacterium, you see that they come back. And if you compare this to a normal, it actually is a little bit higher, but you can see that one bacteria moved into a germ-free mouse. This is called a gnotobiotic, that you know what's living inside of it, can restore an immune cell subset, which is very interesting. So another example that's enumerated above was susceptibility to infection. So they had this model where they took germ-free mice and compared them to conventionally raised mice. So no bacteria in the intestines, bacteria in the intestines, and they infected them orally by feeding them salmonella, which is pictured right here. So what they find after they do this is that through an oral route of infection, the germ-free animals were much more susceptible to lethal infection with salmonella compared to a normal mouse. And the LD50, in this case, is a measure of the dose of bacteria that would be lethal to 50% of the animals. So you can see, in this case, the germ-free mouse has 1 million times less bacteria that is required for it to be lethal in this circumstance, suggesting that gut bacteria normally can have host protective functions. And perhaps the most interesting part about gut bacteria are the metabolic changes that are associated with them. So what we know now is that animals that are resistant to diet-induced obesity when they are actually germ-free. So how you have models where you induce obesity in mice is you feed them a high-fat diet, like, say, a burger, for instance. It's a bit more controlled than that, but you compare them to a conventionalized mouse right here, which is obese, and then a germ-free mouse. They're fed the Western diet, which is basically a high-fat diet in this circumstance. And here is a graph of weight gain over time. You see that the germ-free mouse does gain a little bit of weight, but very, very little compared to a conventionalized mouse, which suggests that the presence of bacteria in the gut has some role in metabolic dysfunction. So I've shown you all these mouse studies, but you may be wondering, so that's great, but you're not germ-free, so why should you even care, right? Because humans don't exist in that germ-free setting. So the curious thing about this obese mouse, actually, is that when you take their fecal samples, you can find that there are differences in the distribution of the bacteria, like phyla or groups within their intestines. So basically, what we see right here is that when you have an obese mouse, it has a difference in its microbial communities that exist. So one cool experiment that they tried with this observed difference in the bacterial composition between an obese and a lean mouse is taking germ-free recipients, so they both have no bacteria present within them, and they feed them either the microbiota of an obese mouse or the microbiota of a lean mouse. And what they find is that after giving them normal diets, not a high-fat diet, they see that the obese mouse, the microbiota, leads to generating an obese mouse, and the lean microbiota preserves a lean mouse, which is very interesting because they've linked these changes to the capacity for energy harvest from food, which suggests that the obese microbiota can actually harvest more energy per like gram of food consumed compared to a lean microbiota. So you might also think about, well, that's a mouse. What about humans? We're obviously very different. We live under more varied circumstances, but they've been doing sequencing studies now on human feces of lean and obese individuals, and what they observe is that there's a decreased species diversity of bacteria in an obese individual compared to a lean individual. And proportions of certain bacteria groups are actually associated with obesity as well. So we don't have the direct causal evidence that you would see in the mouse system to say that bacteria can cause or be associated with human obesity, but we have the first piece of evidence which motivated the germ-free study that would suggest that the presence of certain types of bacteria could be associated strongly with obesity. So just to go over this, there's a lot of information in the first couple of slides, so I'll just have a nice review. Intestinal microbes predominantly live in the large intestine. Germ-free mice are a system to study host micromutualism. And microbes can affect health and the immune system, infection, and obesity. So with this generalized review, now's a good time to ask some questions. All right. Okay. Well, What's difficult about this is that our current approaches to studying bacterial communities in the gut are not terribly high resolution, in the sense that you can identify up to a certain level of the type of bacteria, but you can't identify individual species using these methods. So what you'd be doing is you'd be introducing a hammer approach in there, and you could actually do more damage than good in that context. Yep. So the two types of bacteria that were out of whack would be Firmicutes and Bacteroides. These are two bacterial phyla that are associated also with human health as well. Well, the difference between the two is that they're both gram-positive bacterial species, but the exact metabolic functions they do is not exactly 100% known, because, as I mentioned before, the resolution for finding out certain species that would be involved in metabolic function is not currently there at present. But these are associations that have been made so far.
How about the intestines of mice versus humans? Is it identical? Because it's apples and oranges or apples and apples?
So about the intestines of mice versus humans, they are not identical. It's not really apples to apples. It's more about trying to argue basic principles of how things can work within a model system, and then we can use these basic principles to derive and test hypotheses in human systems. Except that with humans, we can't do studies to determine mechanistically how something functions, because there are no germ-free humans to work on, and there's ethical barriers to that.
But these are extrapolation?
Yes.
Is there a path that communities would change if the diet was changed because of the microbiome? And also, is there a difference between vegetarians and
So with the how fast it takes for the microbiome to change associated with diet, and if there are changes associated with vegetarians and, say, carnivores, actually diet can change rapidly the microbiota. There was a recent publication by one of the professors over here, Peter Turnbaugh, that was investigating this. If within a few days, actually, you can change your microbiota by having switching from a high-fat, high-sugar diet to a more like fiber-rich diet. You can see fluctuations that occur.
Take one more question.
Yep.
What sort of sterile food to give to germ-free mice to ensure that they remain germ-free?
So how we maintain germ-free status? So the all the food and water that goes into these mice is autoclaved, so it's sterilized before, and it's passed through an airlock system in order to get into there, and decontaminated through that. And there's stringent controls to make sure that you are staying germ-free throughout the duration of their lifetime. Okay. All right. Well, also, conveniently enough, we have a little bit of an intermission right now, so we'll take a little bit of a break, and then you can ask me some more questions as well.
Yeah, Chris will be down here to answer some questions.
Any other like behavioral or cognitive differences in the mice in the germ-free environment versus, I mean, have they?
So there might actually be some connections to this. I'm not necessarily sure with the germ-free, but there were some studies about gut permeability and intestinal dysfunction with autism-like behavior that happens within mice. I believe that also happens within germ-free.
There's a new study out that says autism and intestinal dysfunction are related.
Yeah.
Most of the reporting there is we don't think autism caused the intestinal dysfunction. But then nobody sort of ran it the other way, which was intestinal
Function cause autism. Yeah, I think the jury's still out on that, but what they have is for that model is this maternal immune activation model, where they basically have a pregnant mother and they inject it with a immune stimulatory agent. So then the mother has an inflammation period when when the pup is in gestation, and they find intestinal permeability issues with these mice, and then associated behavioral deficits. I believe that might happen with germ-free mice, but this is done in the conventionalized setting. But there are some connections right there. They could correct it actually with one of the bacteria that I'll talk a little bit about later, called Bacteroides fragilis, which seems to promote a more healthy gut. And what it did in that instance was it corrected the barrier functions of the gut, so there was associated changes in the nervous system. But there's a lot of information about how the nervous system innervates the gut and how that can change behavior. It's really unknown at this point.
It's curious that you mentioned that they have very, very low levels of H17 cells. And would you see a higher incidence of spontaneous autoimmune diseases, or do you actually need to do a little more manipulation to induce the disease?
So actually, at least initially, Th17 cells were associated with autoimmune disease. Now we know them more as agents of barrier immunity and immunity against extracellular bacteria. And in this case, since there's no challenge, let's say, to a barrier surface from a bacterium, there's no need necessarily to have a barrier immunity set in place, per se. But at least there are no instances of spontaneous autoimmunity in the germ-free setting. But if you take autoimmune models that are predisposed to this, you put them in germ-free settings, you see that there's a less penetrance of disease. So there's this model of multiple sclerosis called EAE. It's an experimental autoimmune encephalomyelitis, which means brain inflammation, basically. So it's brain and spinal cord inflammation. And what they find is they have a spontaneous model where if they're conventionalized mice, so they have bacteria in their guts, they have an 80% penetrance of disease, which means that 80% of their mice experience paralysis from their brain inflammation. But when you take the same strain, you put it in germ-free, 0% get disease. Yes, it's the other way around. And actually, I'll just put some examples of this later in the talk.
Do mice have rheumatoid arthritis? And if they do germ-free mice get them?
Germ-free mice do not get them.
Do not have germ-free mice do not have rheumatoid arthritis.
Yes.
But germ-present mice do get arthritis.
Yes, if you have susceptible models. So actually, this is what we'll be talking about later as well. That bacterium that I talked about, SFB, which can engender Th17 cells. It's thought that Th17 type cells are associated with autoimmunity and RA.
They're just an activation.
And actually, when you take a germ-free mouse and you colonize it with, a germ-free mouse that's predisposed to arthritis, you feed it normal, species from a regular mouse. And then if you also feed it SFB, you get SFB, that bacteria that I talked about before, that can increase the Th17. You can see that you see more arthritis disease in this model. Yeah.
We're going to get started again in about a minute.
I had curious if you guys did any studies on environmental factors changing the microbial ecosystem in the gut.
Well, environmental factors are probably mostly linked to diet.
I meant more like temperature, activity.
Well,
I was talking to a friend and they're like, we know that we get sick when the changing of season happens.
The changing of season happens?
Yeah.
So,
you guys do any diet tests, maybe to do a test or something like that?
So, that may not necessarily have to do with the bacteria in the gut per se, because since that's in your core, it stays very normalized with the temperature. Your core body temperature typically doesn't really drop below a certain threshold of 98.6. If it does, then you have a lot of other problems besides just the bacteria. But there's been some studies about how colder temperatures can actually compromise immunity in, say, your nasal tract. So it can be actually explain why, when temperatures change, why you're more susceptible to certain colds. So there's some cool work about that as well. But yeah, there's a physiological basis to why you can get sick during certain times of the year.
Just borrow your last one. Nah.
Am I okay on time, or should I go a little bit slower?
I think you're okay on time.
Okay, because we're supposed to.
We didn't even anticipate the intermission being so soon. But it's like,
That's fine. I mean, but the thing is, I have two long things, and
that's okay. And you'll stop.
yeah. Oh.
All right, everyone. I think we're gonna get started again.
Oh. All right, fine. Hey everyone, I think we're gonna get started again. Oh, I didn't have the mic on.
So thank you all once again for coming. One thing we wanted to mention to you, if you check out our website, there's actually a link to a live stream. We broadcast each of our lectures live, and you can actually ask questions live as well. So say hi to everyone on the live stream. So if you're unable to make it in person to one of our lectures, feel free to tune in live, and these will also be posted on our website later on. So with that, Chris, take it away.
All right. Hope you all had a nice intermission. And so I'm going to return back to the plan of the evening. We went over a few of the basic definitions about like the microbes and where they live and why they're important to health. So a curious question now is, how do we study these bugs? And what am I researching? So methods to study gut microbes have really been rooted in the ability to culture these microbes, right? But there's a really big problem with this though, because you can't really know the nutrient requirements for something that you don't know what it is, right? It's kind of a shot in the dark, right? So some things would grow, some things wouldn't. And it happens that about 70%, or even more actually in some cases, of your gut microbes are unculturables. You can't culture everything. So you're really missing a lot when you go with this kind of approach. And another thing too is, when something grows in culture, does not indicate if it's actually numerous in its original circumstances. Essentially what you're doing right here is you're enriching for the weeds of biology. So this bug right here may not really even be that abundant within the intestines, but culturing would say it is. So you need a better method for this. We have a solution. It's called DNA sequencing. So why do you need a sequence to identify bacteria? Well, as I mentioned before, most species can't be grown in culture. And there is competition between species such that this would be another influence to say that what you try to grow on a plate might not necessarily be there when you have a normal sample. And also this provides very broad coverage. Instead of having maybe a few microbes that you can grow on a dish, you can get actually thousands of different identities mapped with this kind of procedure. So how do they figure out bacteria communities from poop? Poop's a very convenient sample to use for these circumstances because you can actually extract lots of bacterial DNA. Well, I was going to say another word, but from poop. And they do this actually through sequencing these thing called the 16S ribosomal RNAs, which are encoded in the genome of bacteria as the DNA. So these things are common throughout pretty much all life as a ribosome because it assembles proteins. Proteins are, basically your workhorses of biological function. So there's actually a shared components between bacterial ribosomes that you can use to map relationships. And here's a diagram of RNA. If you're not unfamiliar with it, it's it's a relative of DNA, and it's also it's a nucleic acid as well. So how do you reconstruct these relationships from genetic information? Well, first of all, let's take a step back and introduce you to the man who kind of pioneered this technique. His name is Carl Woese. This charming old fella. He was actually responsible for identifying a whole domain of life, archaebacteria. But people didn't believe him for a long time, but they were actually wrong. He did prove that there was a whole domain of life, like right here, it's called archaebacteria. And here is a tree of life with different types of bacteria and archaebacteria, and then eukaryotes present right here. So what you can use actually is there are certain sequence differences between the 16S ribosomal RNAs that can identify certain groups. So this is called phylogenetics, being able to reconstruct relationships based on genetic information. So for instance, what you could read for a firmicutes could be very different for a proteobacteria in this case, and then you can give distributions thereof. It's not 100%. As I said before, the resolution doesn't give you the species level, but it gives us at least a broad picture of community structure that happens. So we call this area of study metagenomics because it means above the genome. And it can measure differences between your DNA encoding ribosomal RNA sequences. And also these differences can imply different lineages in bacteria. And most importantly, you can identify unculturable microbes. So the funny thing about this technique actually is that it really wasn't meant to study the human gut per se. It was actually meant to actually sample the environment, like volcanic vents and seawater, areas that you can't really get a good picture of the community structure by culturing approaches. So it was actually a nice translation to take this technology and apply it to human health. And we can now understand, uncover and unravel the mysteries of the microbiota. So there are a few things that we can learn from these sequencing-based approaches and how they monitor the bacterial communities living within us. So first thing, sequencing can reveal the abundance of certain types of bacteria. So you can base this off the number of reads that you get per gram or milligram of sample that you have. So in this case, you can see that in a mouse that was not treated with any antibiotics at all, you can see that there was a high read of bacteria above 10 to the 10th. So there are 10 billion reads that were taken from this from the sample. But whereas when you give it various antibiotics or combinations thereof, you see that this number decreases, which implies that you decrease bacterial load when you give antibiotics, which makes sense. So what this technology can do is it can tell you how much of something you have. And also, it can tell you a general picture of what you have. So this is each color represents a certain bacterial lineage. And you can see that when you have mice that are not treated with antibiotics, they have a certain, variable, but still partially conserved picture of their bacteria. But when you give them antibiotics, it changes pretty drastically, actually. And these are, broad changes that happen with this perturbation. So we know that it can give you abundance and community structure. So another potential method, as well to investigate the microbiome, is called whole genome sequencing, to actually look at more than just the identity of a bacterium. So basically, in this process, you don't necessarily just amplify and look at one particular gene or in the bacterium, but you sequence all the genetic information present within feces. So what this actually has shown is that there are shared genes amongst diverse microbiotas. So this distribution map that I showed earlier, it's highly variable from individual to individual. But when you do whole like genetic information sequencing from that, you find that there are shared genes that exist. So in a sense, you can have different microbes so long as they're doing the same job. It doesn't necessarily matter so much. And this established the idea of a core microbiota in the sense that you can have one bacterium doing this job, and another one can come in doing the same job, and it's basically the same effect. So individual species can actually be less important than the gene content present within a microbiota. So that was a lot to swallow. So if you have any questions right now, now's the time. Yes.
I have a question. In your slide that you showed the untreated pieces of the mouse, and then the supposedly recovered after the antibiotics, it looks like you don't really recover your original microbiome even after recovery. Is that true?
Yeah, well, this is actually still rather new research. When you have this like antibiotic perturbation, you might not actually get back to the same level unless maybe you're given a special conditioning regimen to get back to it. Because conditioning regimen to get back to it, let's say. So for instance, there are cases of antibiotic-associated ulcerative colitis that happen. It seems to be a risk factor, actually, for development of disease where prior administration of broad-spectrum antibiotics can lead to disease. It's not fully causal yet, but it's associated at this point. But it might be hard to get back to your baseline after such a strong perturbation. Yes?
What is cryptosporidium, and why did it come down to it after being treated by antibiotics?
So I am not, sorry, what is cryptosporidium and why did you come down with that after treatment with antibiotics? So I am not exactly sure what cryptosporidium is, but
From untreated water.
From untreated water. Okay, but there's this phenomena that is starting to become more understood called colonization resistance in having your microbiota, let's say, to protect you from infections in the sense that there is competition for resources in your intestines, and it's not just you fighting off against this potential harmful microbe. It's other microbes living in the gut that compete for resources in an environmental niche. So eliminating those microbes can actually be harmful and have you more susceptible to infection, which could also explain a reason why the germ-free mouse studies with the salmonella infection, why that is so lethal in their circumstances compared to the mouse that has bacteria present within its gut already.
So my understanding is that you sort of are born with this bacteria. Is that true?
So you're not necessarily born with this bacteria in you in the sense that when you're in the womb, your gut is sterile. But your first exposure to microbes comes when you're born through the vaginal canal. So there haven't been studies that can really look at differences between children born by cesarean section or vaginal birth and differences in their immune system, but this could be something that is an active area of inquiry right now.
So microbes in every individual is not probably what they like.
Yes. I mean, it may be one of your most important in determining environmental factors because it is such a large surface of your body that is constantly coated with environmental influences, let's say. One more question?
For certain medical procedures, if you use antibiotics and you think you're germ-free, like wipe out everything. That's not true.
It's not
you might
true.
go down by thousands, but they'll be possible. You never kill everything.
Yeah, so...
Like the vancomycin?
Yes.
And what's
ampicillin. So in this instance, so the question was that when you go undergo certain surgical procedures, you take antibiotics and you believe that you're germ-free, but you're actually not. So in this case, a broad spectrum cocktail with metronidazole, which kills anaerobic bacteria, neomycin, which kills gram-negative bacteria, which is one of the major classifications for bacteria, and vancomycin, which kills gram-positive bacteria, the other type. This kills most of the bacteria possible, but you still see that it only decreases it by maybe 1,000-fold, like you said. So it may get rid of the levels of certain bacteria that are needed to establish a functional infection, because just having one bacteria isn't enough to get an infection for the most part. You need a few thousand at least. So if you decrease these levels down below a threshold, it's less likely for you to be infected. Okay. All right. So nice to talk about infection. After we've been talking about these microbes involved in health and immunity in this case, you might be thinking, well, that's cool, but aren't bacteria really associated with disease? At least that's what we kind of thought about for the longest time, from isolating the causative microorganism for tuberculosis in like the 1900s or so by Robert Koch. We have always kind of thought about bacteria as causing infections and being harmful to health. But that's sort of really not true for the most part. Like, so for instance, there are some enteropathogens like salmonella in this case, or cholera, that can cause disease. But for the most part, we have 100 trillion bacteria on us, and they don't cause disease. It seems like this is a natural, healthy state when you have these bacteria in you, suggesting that the vast majority of microbes are either innocuous or in many cases maybe even beneficial. So maybe we need to reevaluate our outlook on bacteria. And one of the reasons why they are beneficial is they can actually be controlled by the human systems. So there was some elegant work that was done that actually shows that you can actually spatially segregate bacteria from your intestinal wall under normal healthy conditions. So what you can see right here is that this is a normal mouse, and this is a mutant mouse for this protein called reg3 gamma. So it's an antibacterial protein. It can kill bacteria. And what they find actually is that there's this thing that was called actually the demilitarized zone that exists between bacteria present within the inside of the gut and your host cells that are that form the intestinal wall. And this is about 50 micrometers, so which is the span of multiple bacteria. So it's a pretty large wall. But when you're missing this antibacterial protein, you can see that this wall doesn't exist in the small intestine in this case. And you see that there's bacteria interacting directly with the wall, and it has some negative consequences. So I like to call this understanding bacteria in context, vilifying bacteria, where we look at them in the intestine as something that could be more beneficial under certain circumstances. So back to those DMZ deficient mice, you can see that they actually have a higher level of immune activation in the intestines when they don't have the barrier. So what they observe here, this is a measure of an activated immune system, let's say. And this mutant mouse actually has a much higher level of activation compared to the normal mouse. But this can be corrected by antibiotics, which is suggesting, in this instance, that this immune activation is really due to bacteria interactions with the host wall. So we also know that some bacteria in the gut can actually promote immune homeostasis and can be beneficial, actually. So here is a measure of an anti-inflammatory cytokine. So this messenger, it promotes an anti-inflammatory reaction under most circumstances. And what they find is that a normal conventionally raised mouse has a normal level of this cytokine. But when you have a germ-free animal, it actually has much less. And it's not exactly clear why. But when you take this bacteria called B. fragilis, so it's a Bacteroides fragilis, which is part of that group that I mentioned earlier in the talk being associated with obesity. and decreases in this population were associated with obesity. You find that actually you can restore this anti-inflammatory level in the intestine. And curiously enough, this bug that can restore this anti-inflammatory level actually secretes a compound called polysaccharide A that can mediate this effect, because when you have a mutant of this bug that does not express polysaccharide A, it doesn't rescue this effect, which suggests that actually a bacterial product in the circumstance can mediate anti-inflammatory change in the intestines. So this leads to this slide here, where they have a model of intestinal inflammation, and where when you have an inflamed intestine, you have weight loss as a consequence. And what you see here is that your untreated mice without the intestinal inflammation just stay normal, because they weren't perturbed. But when you treat them with the intestinal inflammation agent, you can see that they lose a lot of weight very fast. But they had 2 dosing regimens for just delivering this polysaccharide A to these inflamed mice. And what they find is actually both regimens can partially correct the inflammation that occurs in the intestine, which suggests that having beneficial bacteria that could make beneficial metabolites, or even giving beneficial metabolites themselves, or products themselves, can actually promote a healthy gut. But there's a problem with this also. Some microbes do have a dark side, though. And there have been some connections between certain microbial populations and autoimmune disease. So, for instance, we have 2 mice to consider. There's a normal mouse and a mutant mouse. Well, it's a green. It's very scary. And what they do with this one is you co-house them together. So you make this mouse live with this mouse. They're different genotypes. One's normal, one's mutant. But after they live together for a long enough period of time, they start actually sharing a little bit of their microbiota. That's because mice have a tendency to eat feces. Humans, not so much. But there has been evidence to suggest that you share a similarity of your microbiota with your family, and especially close relatives. And actually, I think you share your most microbiota with your mother. But that may also depend whether or not you were born by cesarean section or not, though. So we don't know. So what they did is they took these mouse that they co-housed for like 1 month, and they subjected them to this intestinal inflammation model, like I mentioned earlier, using a little bit of a different compound, but it's the same generalized outcome. And what they find actually is that when they take a normal mouse and house it with this mutant mouse right here in this box, you see that they actually experience really terrible disease in this model, as read out by weight loss. Whereas if you have a normal mouse right here, it loses weight, but not to a really significant extent. What's curious about this is that this is a normal mouse, just houses a mutant mouse. How does this happen? And it's like the disease seems to be transmissible in this context. So that argues for the transmission of bacterial species from this mouse to this mouse that can cause this effect. And in this paper, they actually went into this and identified the species that was associated with this as well. I was just showing the main phenotype, or the main finding they had from this paper. And also, it's now known that gut bacteria can actually be associated with certain types of autoimmune disease as well. So how we were able to test this is by taking a germ-free animal, and you can actually feed it normal or test feces, in the sense that you have normal feces from a conventionalized mouse, or feces that can be either monocultured with, say, that bacterium I mentioned before, SFB, or some other bacteria as well. And what they find actually is that when they took these mice and fed them with feces containing segmented filamentous bacterium, the one that I mentioned earlier, that in an arthritis model, where ankle thickening is one of the main readouts, that the SFB containing feces actually exacerbated disease in this model. And if you have a normal germ-free mouse and that's predisposed to developing arthritis, it doesn't actually develop arthritis with very strong pathology. But it seems like this bacteria present within here can accelerate disease. So back to our plan for the evening. We already went over microbes, where they live, the health implications, poisonous bugs, and then finally, what am I researching? So it seems a little bit tangential. I'm an immunity and cancer researcher. So why am I talking about the intestines? So at first, actually, I was working in autoimmunity. And it looks like, actually, that immune responses to cancer, per se, and autoimmune reactions share some level of similarity in the sense that you normally have tolerance to self, where you don't attack self. But in the realm of cancer, you want to attack a certain part of self because it's actually a much cleaner and efficient way than, say, conventional chemotherapies. So we also know that microbiota can educate the immune system, as we mentioned earlier in this talk. And I'll show you a few basic principles about how the immune system can control cancer. And what I'm principally interested in are the microbiota's effects on cancer. And here is a graphic of an evil crab being cancer, of course, if anyone's familiar with the zodiac. So I'll start with some basic clinical evidence about the immune control of cancer in this. So we know that human immune responses to cancer can actually have different prognosis depending upon the immune cell that's involved. So this was done by looking at the level of certain cells called T cells that infiltrate a tumor around the rim or in the center, and with the prognosis a patient has with their T cell counts. So what this graph shows is a disease-free survival over time, disease-free survival being a measure of the time it takes for you to have a recurrence of disease or perhaps, unfortunately, a mortal event. And what they find right here is that the association of having high T cell infiltration into a tumor is a good prognostic factor compared to having a low T cell infiltration to a tumor. And actually, when you look at conventional staging for cancers, there's various stages for it, like stage 1, 2, 3, and 4, that the infiltrates of T cells could actually be better predictors in this circumstance than conventional staging. But that doesn't mean that there's always a positive immune influence on cancer, because different cell types, let's say like macrophages, and say lung cancer in this instance, can have a negative outlook on survival when you have more infiltrating within the tumor. So you see high macrophage tumors have a lower survival than a low macrophage tumor in this circumstance. So it's a very complex interplay that happens with the immune system in cancer. But what we're interested in is how microbiota educates the immune system and how this can shift the balance one way or another in this circumstance. So there's been some recent studies about microbiota in response to therapies in cancer models. And what they initially found is actually that chemotherapy efficacy can be affected by broad-spectrum antibiotic use previous to tumor induction and chemotherapy in this case. So what they find here is that when they don't give antibiotics, well, this is a survival curve as well, they have seen much more efficacy of their chemotherapy compared to when they do give antibiotics in this setting. And they tried another therapy that revolves around the immune system, and they saw the similar effects actually as well, suggesting that there's some factors involved from your microbiota that can change the immune system, and in this case, change how you respond to a particular therapy. So the black groups, these are the ones that were not given antibiotics at all. So they were just given just controls.
the green curve is people.
No, these are mice, not people.
What the green mice were given a different antibiotic?
No, they were given, so the green mice were not given antibiotics at all. And then the orange mice received a broad spectrum cocktail of antibiotics. So the black mice were mice that did not receive chemotherapy. So my main question, since I'm actually a lung cancer researcher, how can modifying intestinal bacteria actually change treatment outcomes? And we do this by actually studying perturbations to the microbiota in several different ways, by using, like, say, genetic models, antibiotics, and models of intestinal damage. And our eventual goal, hopefully, would be to try to figure out ways to productively modify the microbiota to enhance an anti-tumor response. So hopefully we can go from this to this within our studies. So this is a very new area of research that's less than a year old. So we're still developing basic principles of how this functions. And so this brings to mind potential for these microbes as therapeutics to treat things like bacterial infections, inflammatory bowel diseases, autoimmune disease, and then what we're interested in is possibly, with a question mark, cancer. It may be involved, may not, we don't know yet. So now I'm going to go into my final recap of everything that we went over tonight. So first, we know that microbes are important for health. Second, we know that sequencing approaches have really enabled us to study these bacteria. And we know that now that these gut bacteria can actually affect many, many different types of disease states, from autoimmune disease to possibly cancer, and then also maybe even in some cases some neurological diseases as well. The jury's still out on that. And the last thing we know is poop is more interesting than you can ever imagine. All right, so that concludes this Science in the News talk, and I'd like to thank everyone and our sponsors for. Yes?
What are fecal transplants? How do they work?
So fecal transplants are really a hot topic of research now, because they've been used to some remarkable success, actually, for treating Clostridium difficile infections. So C. difficile infections are associated with antibiotic use, and they're very hard to treat. I mean, one of the main ways to treat them is by using dosing regimens of vancomycin to get rid of them, but that has limited efficacy. And there was a small trial by a Dutch group where they tried fecal transplants, where they took feces from close relatives or next of kin, and they were able to deliver these microbes in a special slurry through an enema in order to reconstitute their microbiota. And it actually showed remarkable success within the small study. So it's unclear exactly what you're giving back to them and what effects it's having, but it seems to be beneficial under many circumstances. Yes?
Do you think we're going to come to a time where you'll be able to go to a pharmacy and get, like, probiotics for your particular condition?
It could be possible, but it's such a complex system where it's unclear if going to a pharmacy to get the probiotics that you need to help cure a condition will actually materialize, in the sense that we need to first identify, I guess, what kind of core microbiome you need for health before, and then see if we can stably make that, in a sense, or put it together before it can be commercialized. Yes?
Two things. One, I would be surprised if there wasn't a fecal bank at some point where you could store your
take antibiotics, and you might need your bio before you go, after you're done.
Mm-hmm.
That's one thing. Two, has anybody studied stress? How it affects gut bacteria?
Those are good questions. So the first question was a fecal bank for storing your microbiota before undergoing, say, antibiotic treatments. And the second was about how stress affects your gut bacteria. So the fecal bank sounds like an interesting idea, and I'm not sure if that's in current clinical practice. But I don't really know how stress affects the gut bacteria. Let's say in a mouse, it may be easier because you have more controlled settings, and you can control diet because typically when humans get stressed, they go to a drug called vitamin chocolate, which has high sugar in some instances, or you tend to eat more high fat or high sugar when you're feeling stressed. So there are a lot of confounding variables that could exist when studying stress and effects on gut bacteria. But at least most of we know about what affects gut bacteria directly is related to diet at this point.
From the interaction with gut bacteria and therapy, is there a hypothesis how this would work? I mean, does the drug end up in the gut somehow and get changed there, or does the gut bacteria release certain receptors that end up in the bloodstream and interact with the drug, or where's the
So there were 2 papers released on this thing about how chemotherapy and antibiotics are affecting one another. First, the hypothesis is that certain factors released by your gut, which was not exactly clear in the manuscript published, can make your immune cells less capable of producing certain inflammatory cytokines that can be anti-tumor, in a sense. So there was not a distinct mechanism of how this occurs, but it suggests that the basic tone of your immune system can be controlled in part by what's living inside your gut. There have been some studies actually suggesting with models of influenza infection where treatment with broad-spectrum antibiotics before infection with influenza can have a depressed antiviral response. So we know that whatever you have within your gut can have systemic effects actually within the whole body. So at least there's some basic principles laid out right there, but the exact details of how it works are yet to be determined.
On the models of the mice with the inflammation,
Mm-hmm.
and there's assumed that they were losing weight.
Yes.
Was that also evident in the earlier mice biome studies that you've been talking about where they were different from these mice?
So in the intestinal inflammation models with the weight loss versus the obese mice, in those studies there was no weight loss known associated with the bacteria or microbiota in that circumstance, but they weren't subjecting them to this particular model. So how they do this model is that they give the mice a special agent in their drinking water, which induces disease. So the disease is only evident when you start it. So if you don't start it, you don't see any effects otherwise.
Is ingesting bacteria, does that work, or is stomach acid there really going to stop us from eating healthy bacteria and really leave us only with fecal transplant as a way to get bacteria to where they need?
So does ingestion of bacteria actually help or hinder seeding of transferred materials? That is an interesting question, and it depends on the type of bacteria. Some are more acid resistant than others, but one of the main impediments, I would say, to having a, productive transplant, let's say, or in some cases even an infection, would be this phenomena called colonization resistance, where there's competition for resources within the environmental niche of your gut. and the people already pre-existing there, or the bacteria in this case actually, will prevent the newcomers from moving in and setting up shop. So it's kind of like you already have a house, right? Someone else tries to move into it. You're not going to let them move into that house, right? You're going to fight them for it. And that seems to happen actually in certain cases, perhaps to prevent productive infections from happening in your gut. And also, on the converse, that could prevent engraftment of a transplant, hypothetically. And it also could be one rationale as well for why you have certain diseases and ailments that can be associated with antibiotic use, because when you kill off these bacteria that are responsible, say, for colonization resistance, then something else from the environment, or if it's living naturally within you at low levels, can outgrow and then cause an infection or other disease. Yeah. Yes.
This is very interesting that you showed that effect of the antibiotics on response to chemotherapy and the immunotherapy. I don't see how that could be shown in humans, but would the ramifications be that perhaps if somebody, I can see that anyone would perform a cancer surgery without antibiotics, but would it be reasonable to not start chemotherapy until maybe introduce a longer period for the person's bacteria to recover?
So in reference to the chemotherapy and antibiotics study, the system they used for this, in my opinion, was rather contrived. What they do establish, though, is a general principle that bacteria in the gut, let's say, can affect a response to therapy. That doesn't mean it's directly applicable to humans at this point. What they did in their system was that they pre-treated with antibiotics before even inducing a tumor, and then give it. So it's a very artificial system, but it establishes a fundamental point to say that these bugs can affect a treatment response. But we don't know how it applies to humans yet. Yes.
I'm sorry if you've already covered this. I walked in a little bit late, so I was trying to listen on the live broadcast. It was a little bit loud. So I had a question regarding there's a proof of increase of gut health and wellness with microbiotic in patients with autoimmune diseases, such as MS.
So that so if there's proven healthful increases in certain autoimmune diseases when you have the right type of microbes administered to you as a probiotic, let's say, for example. So that hasn't been demonstrated yet, and there have been studies done for that, but there's no conclusive evidence to say that it has a beneficial effect, from what I recall. And it seems like one of the few things that seems to have some baseline level of response to having a quote-unquote probiotic therapy is ulcerative colitis. But ulcerative colitis itself may not actually be an autoimmune disease, more so than a attack against bacteria that exist within your intestines that are, in a sense, bad or able to cause inflammation. So for instance, like that mouse that was missing the demilitarized zone with the bacteria in the host intestine. So I believe in some inflammatory bowel syndrome patients, like with ulcerative colitis, that this could actually be thinner or even degraded in some cases. But there hasn't been clear evidence showing that there is a probiotic therapy that can be done to treat autoimmune disease at this point. Though there are studies now where you can identify at-risk people for autoimmunity and collect their feces serially over time, and then sequence the feces of individuals that have the onset of disease to see if there are shared signatures that exist from a bacterial point of view that can precipitate disease. So then you can have better targets downstream to see what you can treat them for that could possibly have an impact on their condition.
Given that there's different diets around the world in different environments,
anyone's studied the generally, what the right kinds of microbiota groups people might have. So in Boston, you can have, given their diet, different ways of
So, in regards to the geographic diversity of human microbiota, and if there is like one type of right microbiota, it really depends upon environmental circumstances and needs. So, for instance, one example comes to mind where I believe certain Japanese individuals are able to derive more omega-3 fatty acids from fish compared to people raised elsewhere, which suggests that there is some sort of host diet driven effects on the microbiota. So I'm not necessarily sure if it's due to like your human genetics more so than what is commonly eaten in that given circumstance. Yes. Mm-hmm.
We have a question from the live feed, Andrew in Rochester. He says, you talked about how different antibiotic regimens can lead to specific patterns of gut bacteria. His question is, are negative outcomes post-antibiotic treatment a result of 1, different stable patterns of bacteria, 2, rapid changes in the bacteria composition, or 3, vulnerability to different types of bacteria like epithelial infection? Or is it more simply that the microbiota is educating the immune system, or are the cancer outcomes just a matter of systemic inflammation during therapy?
All right, that's a very long question. So to parse it apart, sometimes when you have, let's say, an infection after antibiotics, it's really possibly because you had an indolent bug present within you that is able to outgrow when there's more room. If it's already naturally antibiotic resistant, that seems to happen with certain cases like C. difficile or vancomycin-resistant enterococcus. And it can also be because you're in the hospital setting and it's something that's readily communicable. So, for instance, C. diff infections are one of the common infections you can get in the hospital, and also staph infections as well. I mean, the famous one is methicillin-resistant Staph aureus. So it can be a combination of factors in that respect. And also in reference to the systemic inflammation and chemotherapy, it's also known that, well, chemotherapy itself, it doesn't just kill host cells. We don't know if it actually can get to bacteria when you deliver it, but there is chemotherapy-associated dysbiosis that occurs, though the effects of it and what it does are rather unclear. But at least some groups have some explanations for why this occurs, but it's not really a solid science, I would say. One last question?
I have more of a comment than a question before, but I had actually really doing the microbiota and the problem behind that is that actually consider that selective happens to be more germ-free. So they're actually higher density of here that we don't have. So it could be possible that the next step that we have in the end is not as possible. So being able to express knowledge.
I mean, that could be possible, but
Well, there are certain types of parasites forms and some called names, but there are several. They are studying that. for system, and also the for other people. These are ones that people have because don't have to really don't want.
Yeah. Yeah. Well. Yeah. There is a medical doctor at Tufts that experiments with, I guess, helminth infections or worm infections and inflammatory bowel disease. And there may be some connections to that, but on the whole, though, for our largely decreased risk of infectious disease, it I wouldn't say necessarily justifies, but it counterbalances the small risk of autoimmunity that we do encounter in everyday life, on the whole. Yes. One more.
So a question on that. My question is that everyone sort of has the same symbiotic niches, and that the specific species control has anyone started to think, okay, let's develop the tech of changing which species play a role, or that kind of manipulative exploration?
So with the, I guess, the manipulative exploration of the microbiome, that is an active area of research. And the hard part about it is getting fine enough tools to dissect what's happening. So one of the main problems that I was saying is that we can't really identify a particular species about and how they affect our health. We can get a broad group classification for them. So until we can really get a good finger on the actual microbe that's causing a certain effect, we can't really engineer at this point. But first, we need to develop. We can start building the tools, but the tools will be necessary to identify what is present within a healthful aspect. So then we can reproduce that and then manufacture it.
All right, thanks, Chris, again. You can leave your filled out surveys up front on the desk, and Chris can take more questions. And join us again in 2 weeks for exploring planet here in Florida.
Sharing actual methods that I have first. Okay. I just have for the tea, so it's a couple questions.